The American Museum of Natural History really first went to the Gobi in the 1920s in a series of expeditions. And their original purpose was to actually find evidence for the origins and early evolution of humans. It turned out that the expeditions were technically a failure, but in the midst of their effort to do that, they basically stumbled upon incredible deposits of dinosaurs and early mammals in the Gobi Desert. We first returned to Mongolia in 1990 with basically 3 paleontologists from this museum, myself, Mark Norell, and Malcolm McKenna. We found enough on that first expedition to really excite us. And ever since, we've been going back to the Gobi. I'm Michael Novacek. I'm the Provost of Science here at the museum, but also a curator of paleontology.
I'm Paul Velazco. I'm a postdoctoral researcher in the division of Paleontology. In 2010 and 2011, I joined the expedition by AMNH and the Mongolian Academy of Science, recovered 2 tritolodonts. The tritolodonts they're a group of cynodonts.
It's hard to describe what a cynodont is. A cynodont, we used to call them mammal-like reptiles because they had some signs of mammalian features, just a few clues in the skull. Now we're more inclined to call them reptile-like mammals, but they lie outside of what we call bona fide mammals. So cynodonts are a sister relative or a sister lineage of mammals. They're very ancient, over 200 million years, and there's some very strange adaptations in these animals.
So these tritolodonts, they were a group of cynodonts. The smallest was the size of a small rat. The biggest, probably, it was similar to a big domestic cat. It's thought that tritolodonts, they lay eggs. They had a worldwide distribution, and they were living alongside dinosaurs. I study bats, and when I study living mammals, I have described several new species, and I describe them with tons of evidence. I had like DNA evidence. You usually have access to big series of individuals from the same species. But when you have a fossil, sometimes you only have one specimen, and not even one complete specimen. So you have to describe every single bone, you have to describe angles, curvatures, and compare that with other fossils. We have to use CT scanning, SEM photography. You have to do your best and try to be as detailed as you can. Some fossils are known just by 1 or 2 teeth.
We call them derisively spare parts. And I've worked on spare parts for a long time. I basically had to learn how to really study teeth in order to work on these ancient creatures.
Just by studying all the shapes and sizes and forms in the teeth, you can have an idea of how the relationship between the groups of mammals came about. One of the most important characteristics that distinguish our specimens from other tritolodontid species resides on the teeth. Tritolodonts' teeth, they are really amazing. That's where the name tritolodont comes about. The upper teeth have 3 rows of cusps that face forward, and the lower postcanines, they comprise of like 2 rows of cusps that face backwards. And they were working like this, to shred the vegetation. The combination of numbers between each row is called the cusp formula. Is unique for each genus. And we found that our 2 fossils are different from other tritolodontids and each other.
I was so excited looking at these tritylodonts with Paul. It appears that these species that we've discovered are new genera. They're completely new lineages.
To just base the whole description or the whole set of characteristics that will define this genus and just based on one specimen, there's more pressure to do a better job.
There's an enormous amount of curiosity inspired by looking at something that's so strange. And in a sense, these things are alien. They're from another world. It's our world, but we're talking about 80 million years ago. And so you immediately transport yourself there because you're saying, well, how did this thing live? What was it related to? What was a day in the life for the Tritylodon? They were very distinctive animals.